Hi, I'm Kyman Savage. I'm doing biology A-level, and today we're going to be going over glycolysis. If you haven't watched my video titled Coenzymes, I'd advise you watch that before you watch this one, because it outlines the whole process of glycolysis and the coenzymes involved and what they do. So it'd be kind of helpful if you watch that before you watch this video. But by the end of this video, you should be able to state that glycolysis happens in the cytoplasm. You should be able to outline the process of it, and you should also be able to talk about how in aerobic respiration, pyruvate is actively transported into mitochondria. So basically, glycolysis is a metabolic pathway, and it occurs in all living cells that respire, so it occurs in eukaryotic and prokaryotic. It's most well understood, it's the oldest known pathway. So, it's known really well, so you know everything that happens in it to a very fine level of detail. The actual pathway consists of 10 stages, 10 reactions, but we don't need to know it in that much detail. We only need to know it in outline, so we only look at it in 4 stages. And the 4 stages are phosphorylation, the splitting of hexose 1,6 by phosphate, the oxidation of triose phosphate, and the conversion of triose phosphate to pyruvate. So start off with stage 1, phosphorylation. Now, glucose is a hexose sugar. By the way, glycolysis, if you just want to know the definition, is a metabolic pathway where each glucose molecule is broken down to 2 molecules of pyruvate. Okay, good. So, stage 1, phosphorylation. Glucose is a hexose sugar, so it contains 6 carbon atoms. Its molecules are stable, and they need to be activated before they can be split into 2. So, one ATP molecule is hydrolyzed, and the phosphate group released is attached to the glucose molecule at carbon 6. So, one ATP molecule hydrolyzed, the phosphate group released is attached to the glucose molecule at carbon 6. So, this glucose 6-phosphate is changed to fructose 6-phosphate. Now, another ATP is hydrolyzed, and the phosphate group release is attached to fructose 6-phosphate at carbon 1. Now, this activated hexose sugar is now called fructose 1,6-biphosphate. And the energy from the hydrolyzed ATP molecules activates the hexose sugar and prevents it from being transported out of the cell. So energy from hydrolyzed ATP molecules activates the hexose sugars and prevents it from being transported out of the cell. And we refer to this activated phosphorylated sugar as hexose 1,6-biphosphate. And this name tells us that it is a hexose sugar, and it's got 2 phosphates attached, one at carbon 1 and the other at carbon 6. That's where the 1,6 comes from. Also, you've got to note that the stage uses 2 molecules of ATP for each molecule of glucose. So each molecule of glucose uses 2 molecules of ATP. That's great. That's stage 1. That's phosphorylation. I know it's a lot of bits to learn, but it's kind of in order. Like, you know, that it goes from glucose 6-phosphate to fructose 6-phosphate and to fructose 1,6-biphosphate. It's sort of structured. They sort of change a little bit each time. Stage 2 is splitting of the hexose 1,6-biphosphate. That's simple. Each molecule of hexose biphosphate is split into 2 molecules of triose phosphate. That's a 3-carbon sugar molecule, each with 1 phosphate group attached. Simple. Stage 3 is the oxidation of the triose phosphate. Now, although this process is anaerobic, it does involve oxidation. Sorry, one sec. Okay, so 2 hydrogen atoms with their electrons are removed from each triose phosphate molecule. So 2 hydrogen atoms got their electrons are removed from each triose phosphate molecule, which, remember in stage 2, that's where the triose phosphate molecules came from. So the hydrogen atoms removed from triphosphate molecules. This involves the dehydrogenase enzymes. If you don't know what that is, look at my other video. Which are aided, really need to look at the other video, coenzymes for this one. These are aided by the coenzyme NAD, which is a hydrogen acceptor. And NAD combines with the hydrogen atoms, becoming reduced NAD. I remember that even though it's combining with the hydrogen atoms, it still says it's reduced. That's just the word. So at this stage of glycolysis, 2 molecules of NAD are reduced per molecule of glucose. Do you remember in phosphorylation, In phosphorylation, for each molecule of glucose, 2 molecules of ATP were used. In stage 3, oxidation of triphosphate, 2 molecules of NAD are reduced per molecule of glucose. And also at this stage, 2 molecules of ATP are formed, and this is called substrate level phosphorylation. So what's substrate level phosphorylation? It's where there's 2 molecules of ATP are formed. Now you've only got one stage left, which is great. See, they're not that hard. You just got to learn them and just go over and remember. In stage 1, this happens, this happens, this happens. Stage 2, this happens. Stage 3, da da da. Stage 4, da da. Yeah. Stage 4 is a little bit simpler. That's just the conversion of triphosphate to pyruvate. And 4 enzyme catalyzed reactions convert each triphosphate molecules to a molecule of pyruvate. And pyruvate is also a 3-carbon compound. And in this process, 2 molecules of ADP are phosphorylated to 2 molecules of ATP. So in the process, 2 molecules of ADP are phosphorylated to 2 molecules of ATP. Great, we've done it. That's glycolysis. But what are the products of glycolysis? What actually is produced by it? Well, from each molecule of glucose at the beginning of the pathway, by the end of glycolysis, there are 2 molecules of ATP. 4 were actually made, but 2 are used to kickstart the whole process again. So the net gain is 2 molecules of ATP. Also, 2 molecules of reduced NAD are produced, and these carry hydrogen atoms indirectly via a shunt mechanism to the inner mitochondrial membranes and can be used to generate ATP during oxidative phosphorylation. Right. The last thing that's made are 2 molecules of pyruvate. Now, this will normally be actively transported to the mitochondrial matrix for the next stage of aerobic respiration, which will be my next video. In the absence of oxygen, this will be changed in the cytoplasm to lactate or ethanol. So in anaerobic respiration, pyruvate will be changed in the cytoplasm to lactate or ethanol. Well, that's great. That's glycolysis, basically. So just learn the stages of glycolysis for ATPs used, ATPs reduced, and NADs reduced, and you'll be set. If you want some questions, outline the roles of coenzymes in the glycolysis pathways. You could also explain why the net gain of ATP during glycolysis is 2, not 4 molecules. You could also explain how oxidation occurs during glycolysis, although no oxygen is involved. Well, thank you very much for watching this video, and good luck with your exams.